I'm going to focus on drug metabolism. So what must a drug do other than bind? So as we kind of discussed before about our ideal drug, an oral drug must be able to dissolve. It must survive a pH range of anything from 1.5 in the stomach to 8 in the intestines. Once it reaches the intestines, it must be able to survive intestinal bacteria, also be able to cross the membranes, survive the liver metabolism, avoid active transport to bile, avoid excretion by the kidneys, and partition into target organ, and also avoid partition into underside places such as the brain and the fetus. When we consider drug metabolism pharmacokinetics, DMPK, this helps to design and select candidate drug molecules with required efficacy and safety, known as ADME. So A stands for absorption, D for distribution, M for metabolism, and E for excretion. So when we think about pharmacokinetics, this is a branch of pharmacology that determines the fate of the substances delivered to the body. And so front-loading of DMPK decreased drug attrition from 40% in the 1990s, now down to 10% after 2000. Now it requires a candidate drug profile which details information about the efficacy, the potency, the safety, and the ease of use. Drug metabolism. So the metabolic breakdown of drugs by a body to detox them is known as the pharmacokinetics. And there are different phases. So in phase 1, we have something called modification. So the enzymes which are responsible introduce reactive and polar groups onto the substrates. And the most common is the hydroxylation by cytochrome P450. And there are also other reactions. So we get oxidation, reduction, hydrolysis, and cyclation. Phase 2. So phase 2 is the conjugation. So this is the activated species in phase 1, which is then conjugated to a charged molecule like glutathione, GSH, or glycine, or glucuronic. This produces larger molecular weight compounds that are less active than their substances and are more polar and so cannot diffuse across the membranes. So for example, if we take paracetamol, so acetaminophen, when it's kind of broken down by cytochrome P450 in the liver, it makes NAPQI, and then you can get the conjugation of charged molecules like glutathione to make this molecule. However, if you get charged molecules which are then added to the protein, you can have the addition of protein, which actually causes liver toxicity once glutathione levels have been depleted. Phase 3 is further modification and excretion. So this is the synabiotic conjugates can be then further metabolized, and the anionic group acting as affinity tags for a variety of multi-drug resistant proteins. Now these are transporter proteins that can remove phase 2 products to the extracellular media. So if we take our toxins, these are like drugs, insecticides, pesticides, pollutants. You get breakdown products from hormones and also neurotransmitters. In phase 1, this is mainly done in the liver, so this breaks down the fatty toxins. So the genes that are involved are cytochrome 1A2, cytochrome 2C19, cytochrome 2D6, and cytochrome 3A4, and more. In phase 2, so this is it binds with phase 1 products, which makes them water soluble. Genes that are involved are for example, SOD1, GSTs, UGTs, and NATs, and more. And then in the last phase is the elimination. So that's through the kidneys in the urine, and also through the skin, through perspiration and sweat, and also through bile and intestines through excretion. Optimization of DMPK properties. So there are various strategies in silico and in vivo that are used to advance drug candidates with attractive DMPK properties. And that includes like looking at absorption, variability, avoiding PK based drug-drug interactions called DDIs, or achieving or avoiding central nervous system exposure, and also clearance. So when we look at the drug metabolism of pharmacokinetics, we're going to look at different aspects. So we're going to look at, for example, drug interactions, the reactive intermediates, the enzymology, the pharmacogenetics, the active metabolites, toxicology, and lead optimization, as well as the transporter and bioanalysis. So it includes a whole branch of things. And the role of metabolite identification is, like, for example, the active metabolites and minimizing the risk for reactive metabolites. DMPK also needs to predict human pharmacokinetics and a safe efficacious dose which ensures biological activity in humans. Absorption and bioavailability. So oral is the preferred route of administration, as we realised from our pre-formulation studies, and we need to characterise oral viability as F. So this is the percentage of dose drug that reaches the systemic circulation compared to intravenous route. F must be optimised, as low F is associated with poor exposure and lack of efficacy. So bioavailability depends on 3 serial steps. So you've got F equals FA, which is the fraction of drug absorbed, affected by the gastrointestinal solubility, so it includes the particle size, the dose, the pH, solubility, and formulation, as well as the effective permeability. So this is passive and active transport, and also the gastrointestinal stability. Fg is the fraction, of the drug escaping intestinal metabolism, and that's affected by metabolic enzymes in the intestinal wall. And Fh is the fraction of drug extracted by the liver. That's affected by the metabolic enzymes in the liver and the transporters if a drug is excreted unchanged into the vial. So looking at cytochrome inhibition, there are 2 cytochromes known as cytochrome 3A4, this one here, and cytochrome 2D6, this one here. And these are based on drug-drug interactions, which are most common and occurs in the liver and also in the intestine. If we look at the effects of cytochrome inhibition, for example, terfenadine, phenadine plus erythromycin, we can get inhibition of cytochrome 3A4 isoenzyme. And what happens is you get with toxic levels of terfenadine, this can lead to enhanced risk of cardiotoxicity. There are 4 main forms of pharmacokinetic drug-drug interactions. The first is competitive reversible cytochrome inhibition. The second is metabolism-based time-dependent cytochrome inhibition. Also, we have uptake and efflux transporter inhibition, and also cytochrome induction. And in fact, there's inhibition of 5 major cytochrome isoforms, including 1A2, 2C9, 2C19, 2D6, and 3A4, which are evaluated first, and the later access isoforms are 2B6, 2CA, and 3A5. And also there's a potential for drug-drug interactions with co-medication, which is reduced if there are several clearance mechanisms involving the metabolism by various cytochrome enzymes. Here, this graph shows the percentage of drugs, So this is like the routes of elimination of the top 200 most prescribed drugs in 2002. And if we kind of focus on the cytochromes, you can see the percentage of drugs which use these different cytochromes. Okay, so if we look at competitive reversible cytochrome inhibition assays, we can use first, for example, a fluorescence assay. The fluorescence assay is cheap. We can use it for high throughput screening and the initial profiling of large number of compounds. So essentially we take a 96 well plate, we add our test compound, an inhibitor, plus our microsomes and also a probe substrate. Afterwards we add a cofactor such as NADPH. And then we can then measure the product formation as fluorescence or using kind of liquid chromatography mass spectrometry, LCMS. The LCMS assay is actually more expensive and you can essentially have lower throughput, but it's more predictable and can be used to verify hits from the fluorescent-based assays. So if we get a fluorescent-based hit here, we can use the mass spec to verify the screen. And if we kind of look at the points of accessing drug-drug interaction potential for new chemical entities at the stages of drug development, so for hit identification, we can have a single point inhibition of cytochrome and use the fluorescence detection, and also a determination of potent versus weak or non-inhibitors. For the lead identification phase, we'll perform IC50 calculations in the cytochrome, also use the fluorescent-based detection or an LC-MS, and then also maybe look at IC50 to extrapolate the apparent Ki. It's not until we get to the kind of clinical evaluation that we'll perform a full Ki determination in human liver microsomes, but also we'll look at the cytochrome probe substrates analysed by LCMS, and also look at mechanistic studies and prediction made in vivo conditions. If we can consider membrane transporters, these control the influx of essential nutrients and ions, but they also regulate the efflux of cellular waste and also environmental toxins and drugs. There are two major superfamilies involved in drug transport. The first are ABC, known as ATP binding cassette transporters, known as, for example, P-glycoprotein, or as can be written as PGP from the MDR1. Also, there are the second family is SLC, which are solute carriers. These are known as transporters, such as 5-HT and SERT. And these all kind of act on different organs in the body. So the membrane transporters play roles in the pharmacokinetic pathways, so in drug absorption, distribution, metabolism, and excretion. So the brain is actually separated from the systemic circulation by this blood-brain barrier. So the blood-brain barrier essentially restricts the entry of drug compounds, so multiple drug transporters are expressed here, and it has low permeability. Now the blood-brain barrier is made up of the astrocytes, a basement membrane, and then these endothelial cells. And how do we assess unbound drug concentration in the brain? So we can look at the efflux permeability assay, so MDCK MDR1. So MDCK stands for Madrin Darby canine kidney. So using these kidney cells, we can transfect with this MDR1, which is a P-glycoprotein. And then these can be then cultured on a monolayer, the PDCK cells can differentiate to form tight junctions that actually mimic the blood-brain barrier. And the drug transport can be accessed in both directions. So we can assess from A to B. So from the top of the chamber, so this essentially is a transwell chamber, okay, and then from the bottom of the chamber back into the apical chamber. And we can then measure the permeability coefficient, which is P ap equals dq, which over dt, which is the amount of drug in the basal through A and B, or apical B and A chamber, as a function of time in nanomoles per second. The CO is the initial concentration, and A is the area in centimetre squared. So how do we assess the unbound drug concentration in the brain? So we can use efflux permeability assays. So the MDCK MDR1 assay looks at the sensitivity for P-glycoprotein substrates. Whilst there's another assay that we can use which uses CACUS2 cells. This looks at the sensitivity for several efflux systems, not only P-glycoprotein. Now there's a rule of force for identifying P-glycoprotein efflux substrates. First, we can identify P-glycoprotein substrates. There's a sum of nitrogen and oxygen atoms, which are either equal to or less than 8. It has a molecular weight of less than 400 Daltons, has a pKa of less than 4, and a log P of less than 2. For non-substrates of P-glycoprotein, there's a sum of nitrogen and oxygen atoms, which are less than 4, and a molecular weight of less than 400 Daltons, and a pKa of less than 8. And we can establish an in vitro and in vivo correlation. So this is IV-IVC, using the in vivo experiments to determine the ratio of Cu Br over Cu pi. And the fraction unbound in the brain is determined ex vivo using brain binding assays such as the brain homogenate or brain slice. So assays for candidate drugs requiring access to the central nervous system. So in the phase of drug discovery from hit to lead, we can use these bidirectional COCO2 assays, and we can measure the permeability and efflux liabilities and select hit based on access of liabilities. For the lead identification, we can use the bidirectional COCO2 assay to determine the optimization feasibility, and for early phases of drug discovery, we can use the in silico tools. Are these useful? If so, can you develop the project-specific model and develop series-specific CNS penetrant tool compounds? For late phase, we can then look at the protein-brain binding assay, so in vivo CNS distribution study. Here we're going to look at the free brain concentration and develop a PD model and also an IV-IVC. And for the lead optimization, so we can use the bidirectional COCO2 assay, in vivo CNS distribution study, as well as the in vivo P-glycoprotein inhibitor and knockout studies. And here we'll optimize away from the efflux substrates and continue to build the IV-IVC and understand the impact of efflux in vivo. And when we kind of focus on the clearance optimization, we must be able to determine key elimination processes. For example, we need to look at the optimization of these processes using the human in vitro system or animal systems. And a typical drug shows the following clearance processes. For example, cytochrome P450 has a mediated clearance of around 55% and oxidation and reduction in the liver. Whilst unchanged drug of around 25%, you have non-metabolic clearance. And other metabolism pathways around 20%, phase 2 conjugation, so amidase, esterase, amine oxidases, deaminases, dehydrogenases, UGTs, etc. Clearance is the sum process of all in vivo elimination pathways, and any one pathway can dominate, and there is kind of case-by-case analysis. Metabolic clearance. So we can identify enzymatic source of instability and exclude other clearance mechanisms, so we can get metabolic soft spots. We can identify the substrate for the metabolic enzymes and then modify the drug to reduce or block catalysis. And this is a rational drug design. And if we do in silico prediction of potential labile sites on the molecules, we can then dock the candidates of the cytochrome enzymes. Okay, so this shows an example of how we can dock the enzymes to identify potential substrates and reduce the possible interactions. And screening of those compounds in vitro in rat microsomes and also in hepatocytes help to determine the metabolic clearance. Also, the use of in vivo studies in total body clearance can reveal if the secondary phase processing outside the hepatic system is occurring. Finally, we're going to look at kind of renal clearance. So this is where we reduce the renal clearance in order to reduce total body clearance. And generally, this is achieved by increasing the lipophilicity through the modulation of something called log-P or pKa. Commonly caused by compounds that do not bind well to the plasma protein, so they have low polarity and low log D of 7.1 of less than 1. The lack of passive permeability and a high number of efflux transporter proteins in the kidney, such as OAT or OCTs. Human clearance is predicted from the animal data, so renal clearance equals GFR times FU. So GFR is the glomerular filtration rate times the fraction unbound. And both rat and dog data are used, so acids and citrulline data are best predicted from dog models. Biliary clearance. So ATP-dependent transporters, such as P-glycoprotein or MRP2 or BCRP, mediate uptake into hepatocytes, so liver, and efflux into the bile. Now, bile accumulates in the gallbladder and then is released into the intestine, and drugs may be reabsorbed, known as the kind of enterohepatic recirculation, known as EHC, which occurs here. And biliary clearance is commonly higher in the rat, mice, compared to man and dog. Biliary clearance, so it can result in rapid clearance and low bioavailability. So low systemic exposure and small safety margins. It's difficult to optimize chemically and predict across species. Often a synergistic effect of two transporter systems, so you have uptake and efflux. If in vitro total clearance is well predicted, there's no specific biliary investigation required. A predictor of biliary clearance is amniotic species is the molecular weight optimum of about 475 Daltons in man. A lower molecular weight compounds can be reabsorbed. Conjugates and drug metabolites are often of sufficient molecular weight for biliary secretion. Track progress in rat and dog using intravenous PK studies without the bile collection. You can make use of in vitro hepatic uptake models and also access efflux in sandwich cultured rat and human hepatocytes. So this kind of ends our second session. Okay, so these are some suggested reading. So this is it's a publication from Werner and Misha. It's a pharmaceutical pre-formulation studies in formulation and development of new dosage. And this is like a review, it's quite a nice paper. The second recommended reading is liposomal formulations in clinical use, and it's an updated review. We haven't really discussed this in the presentation, but it's also quite an interesting review. And the third one is drug metabolism in drug discovery and development. This is actually also quite a nice short review. Anyway, that ends our final presentation.